Fungi are not plants. They're not animals either. But genetically, they're closer to us than to trees or flowers. That alone is surprising, but it's only the beginning. What we call fungi today, mushrooms, molds, yeasts, evolved from single-celled ancestors that looked nothing like them. These early organisms didn't grow in soil or forests. They swam. They crawled. And some of them didn't even digest food the way fungi do now. To understand where fungi came from, we have to go back to a time when life was still microscopic and evolution was rewriting the rules of what a living thing could be. The evolutionary origin of fungi begins with a microscopic organism that left no visible fossils, but whose molecular traces still shape our understanding of life. Modern genomic studies place fungi within the Opisthokonta, a clade that also includes animals and certain single-celled protists. This shared ancestry points to a eukaryotic cell that moved with a posterior flagellum and likely fed by engulfing other cells. It wasn't a fungus, or even close. But it carried within it the basic machinery, cytoskeletal systems, membrane dynamics, early regulatory pathways that would later diversify into animals, fungi, and their lesser-known relatives. This organism, probably aquatic, is the starting point not just for fungi, but for the entire branch of life that we ourselves belong to. Within this group, one branch took a direction that would lead toward fungi. These were the Holomycota, a lineage that included primitive forms like Nucleariida and Fonticula. These organisms weren't fungi yet, but they carried the cellular machinery and evolutionary potential that fungi would later inherit. Some of them could crawl like amoebae and some could form simple colonies. In these humble experiments, evolution was laying the groundwork for something that didn't yet exist, a way of life that would eventually change the surface of the Earth. But a fundamental shift had to occur. These early ancestors no longer engulfed food like their animal relatives. Instead, they began digesting their food externally, a strategy known as osmotrophy. They secreted enzymes into their environment, broke down complex organic matter, and absorbed nutrients through their membranes. This change brought a new wave of genetic innovation: enzymes to break down plant cell walls, transporters to move molecules across membranes, and the early architecture for growing as branching filaments, what we now call hyphae. This transition unfolded in two major bursts. The first occurred in the earliest fungal lineages, chytrids, primitive fungi that still retained flagella for swimming. The second came later, in the ancestors of modern mushrooms, yeasts, and molds, what scientists call the Dicaria. In this second burst, fungi developed dense networks of hyphae and complex fruiting bodies. The genes responsible for multicellularity, signaling, and development all expanded dramatically. These were not just small changes. They were evolutionary leaps that would define what it meant to be a fungus. What began as a single-celled aquatic microbe evolved into an entire kingdom that now stretches from the soil beneath our feet to the insides of living hosts. With each genomic leap, fungi gained new ways to survive, expand, and transform the environments they entered. Their story didn't start with mushrooms, it began long before, in silent waters, with a drifting cell that would one day feed the forests.